As described in the previous presentation, genetics is the branch of biology that studies heredity and variation in living organisms. It explains how traits such as eye color, blood type, or susceptibility to certain diseases are passed from parents to offspring. The basic unit of heredity is the gene, which is the segment of DNA located on chromosomes. Uh, Now the genes carry these instructions that determine an organism's characteristics. The principles of genetics, as we had explained before, it is described by Gregor Johann Mendel. Okay, so Mendel had described the principles of inheritance in this 19th century, and his experiments were performed on pea plant. From his work emerged the ideas of what you call law of dominance, then law of segregation, and the law of independent assortment, which we will be dealing in detail. Law of dominance, in which we will see that out of the two alleles, one which will express itself, two contrasting alleles, one which will express itself is dominant, and that which will not express itself is recessive. So that is dominance. Then law of segregation, which where we will see that the gene pairs separate during gamete formation, and the law of independent assortment, which explains how different traits are inherited independently of one another. So we will be talking about all these things, but before that, let us see this already explained what is monohybrid cross. See, Mendel crossed purebred tall. So I had explained already that why the plant which is selected was pear, isn't it? And what he did in monohybrid cross itself says that it is a cross in which only a single character is different. So here the character for white, what is different? Height, height, in which the alternate forms were, say, tall and dwarf or short varieties. Okay, so he did a cross. Which cross? So this is the parental cross. This is the parental cross which he did. He took tall plants and he crossed it with the dwarf plants. Okay, so he took the tall plant flower. So there again we have to understand, some of this is tall plant flower, and this is say this is the dwarf plant flower. So in any one of these, what he will do, he will emasculate. Suppose here he wants to do crossing, so he will emasculate it. Emasculate means cut it. Emasculate. Cut this before maturity. Okay, so that it cannot self-fertilize. So self-fertilization is prevented. Now what he will do, he will take the pollen from this dwarf variety and he will transfer it over here. So this is the crossing. So he is crossing the dwarf variety pollen in tall variety. So what will happen? Fertilization will be there. After fertilization, what will happen? Seeds would be formed. Seeds would be formed, and then after maturity, the seeds would be again planted, and they will see the result. What will happen? Whether the plants which grow out from this, what happens to them? That will be seen. So this is what they will do. So now after this, what we will see, so this is the gamete. What will come out will be small t. Here the gamete which will come out, small t, and these two will fuse and you will get, this is the F1. This is F1. Now in the F1, what he found, that the F1 generations were all tall plants. F1 generation were all tall plants, but he crossed tall and he crossed dwarf, but F1 was all tall plants, indicating that out of the tall and dwarf, which characteristics was expressed? The tall was expressed. So tall was expressed means tall became dominant. So this is the dominant character. Now, which it could not express itself, that became, this is what we call as the recessive character. So this is the first you can say law, and from here one thing we can say, that whatever character is expressed in F1, whatever is expressed in F1, that is the dominant character. Okay, that is the dominant character. Next, this is uh, this is a cross in which this we can say, this is a parent, this is parent, dominant parent, dominant this is a parental generations, isn't it? So we can show it in the Punnett square. So this is gamete which is coming out from this. Again, from here the gametes which will come out will be this, and then after fertilization, so this T, T and T, so in that way they will make the square. So 100% genotypes are all capital T, small t, so all are tall. So this is the principle of dominance. So here what we say, out of a pair of contrasting characters present together, only one is able to express itself while others remains suppressed. Okay, the character that is visible is dominant character, here tall is dominant, and the one which is not visible, that is recessive, here dwarf is recessive character. Now if we further move on, in the monohybrid cross, now F2 generation will form. So F2 generation, how it will be formed? We are crossing the F1. So F1 plants, if we are crossing, so that means we are crossing tall with tall. So here again, from, suppose for this if I consider, what are the gametes? Capital T would be 1, small t would be 1. For here again capital T and small t. So what is the chance of fusion? This is what will happen, capital T, or so capital T, small t. Similarly for this, capital T and small t, and again small t, small t. So here what you find, you are finding that there is one capital T tall, two capital T small t tall, and one small t dwarf. So the genotypic ratio is 1:2:1. But if you see the phenotype, you will find that these three are tall and only one is dwarf. So here the phenotypic ratio is 3:1 is the phenotypic ratio. Now from here, what can we say? So from here it can be said that this is possible. This is possible means which is possible. The dwarf character can appear only if the alleles separate. Otherwise it's not possible. So dwarf character will appear only when the alleles separate during the gamete formation, isn't it? So from monohybrid cross in the F2 generation, for F2 generation formation, where the dwarf character is appearing, from that came the law of segregation. Mendel said about segregation, in which what he said, an organism's characteristics are determined by internal factors which occur in pairs, only one of which can be present in a single gamete. So differently we can say that an organism has two versions of each gene, say capital T and small t, but one of these versions, small t or t, is passed on to the offsprings. That means they have to separate. Okay, or we can say simply that an organism has two versions of each gene, but only one of this version is passed on to the offspring. This is because meiosis separates homologous pairs of chromosomes. This is the understanding which developed later, so that only one of each pair passes into the gametes. Okay, so when we understand meiosis, we will see that how segregation occurs. But the law of segregation is saying that out of the two alleles, one allele will separate during gamete formation, and the paired condition will be restored during fertilization. So this is a segregation, this is universally accepted. For the first one, law of dominance, we will see there are exceptions, but in the second one, for segregation, it is universally accepted. Here one more example we can give you is of cystic fibrosis. Cystic fibrosis. This is a lethal genetic disease affecting Caucasians. Okay. So here, if you see that this disease is again caused by this allele C. Okay. Now here, if you see, this is the heterozygous capital C and small c we have taken. So what are the chances of if the both parents are, that means here the cross is between capital C small c and capital C small c. So this is supposed to the father, this is the mother. Next, what can happen? These are the possible combinations, that capital C if it occurs, then it would be normal. If capital C small c, then carriers, and small c, then the person has cystic fibrosis. Now what happens in cystic fibrosis? Actually, cystic fibrosis is a faulty gene produced where a defective chloride channel protein. Okay, so defective chloride channel protein in the epithelial cell membranes. Okay, so what happens? This, since it is defective, this chloride ions cannot move properly out of the cells. So if chloride ions cannot move properly out of the cells, then what happens? The water movement is reduced. Okay, and if the water movement is reduced, then what happens? It becomes thick, and this sticky secretions, instead of becoming thin, it will become thick, and that normally the mucus is watery in nature, but now the mucus will become thick in nature. Okay, so why? Because of this chloride ion channels being not properly made. So chloride ion channels, why? Who is controlling again this? This is again controlled by this gene. Okay, this gene, which we are saying that C is the gene. So what happens then? The mucus in the airways becomes abnormally thick and sticky, and this mucus blocks the air passages. Okay, it traps bacteria. So what could be the consequences? There could be chronic cough, recurrent lung infections, progressive lung damage, all these things. So this disease is following the Mendel's principle of heredity. So this is a disease which we find, of course, very much limited to the Caucasian tribes of Africa. Next, let us move to the dihybrid cross. So in dihybrid cross, suppose say we are taking, now dihybrid, as I already said, two characters will be considered. Okay, so here say I am taking round yellow, these are seeds. Okay, round and yellow seeds, round and yellow, and we are crossing with wrinkle and green seeds. Okay, so this is the parent generation. Now parents are always homozygous. This has already been explained why they are homozygous. This is why Mendel was lucky. Now if you take out the gametes here, then what we will get? We will get capital R and capital Y. This is the gamete type here, and here the gamete type would be small r, small y. Now if these two cross, we will get what? Capital R, small r, capital Y, small y. This is your F1 generation. This is your F1 generation. Okay. Next, when I do an F1 cross, so you are crossing basically this with this. This is the F1 cross. Yes. Now when we do this F1 cross, now we have to find out the types of gametes here. So basically when we see the chromosomes are like this, no? So here, say when I draw like that. Next, so what are the genes here? Suppose say here capital R, here say write small r, isn't it? Here say capital Y, here say small y. So what are the types of gametes? Capital R, capital Y, this is one. Capital R and capital Y could be one. Next you say capital R, here splitting can occur. So here splitting can occur, and this is what can happen. So what it means that now here what can happen? There could be a break over here. There could be a break over here. So what is the combination you will find then? You will find a combination of this nature, that in the green part are Y. So this is the green part. What is attached? This part is attached. And again, uh, the other side we can show that this is the part and in which the green part can attach. Yes, from here I am showing you that from here this is what can happen. So then what are the combinations? Capital R, capital Y could be one combination. Next, this part is capital R itself, so capital R would be there. But here what is there? Small y. Here small y is there, so small y. Next, this side what is there? Small r, small r, and with it now which part is attached? This part is attached, so this is capital Y, capital Y. And then last what you get? Small r, small r, small y. Okay, this I can make it green so that now you understand properly. So these are the four types: capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So this is actually a phenomenon of meiosis, so which we should know for formation of gametes. So this is how gametes are being formed. So two heterozygous means what will be the possible types of gamete? There will be four possible types of gametes. At last of the slide I will show you a formula by which we can find out how many types of gametes could be found out. So here was the explanation part. So next we can go for the Punnett square. So Punnett square, so those gametes we can arrange it now. So that means say capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Similarly, for another one, say male one also we can one we can say male, the other one female. So capital R, small y, small r, capital Y, and small r, small y. Now here to make a combination, we make a Punnett square of this nature. Okay, and what we do, we show the combinations. So here one set could be your male gamete, the other set could be your female gametes, and when you make combinations, this could be the combinations, no? Capital R, capital R, capital Y, capital Y. Similarly, for this capital R, capital R, capital Y, small y. Again, next you could go with this and this. So again, capital R, small r, capital Y, capital Y. So in that way we can fill all this up and you get a combination of this nature. Okay, so these are the gametes, two gametes, yes, and these are the combinations. So one set we can say this is the male and one set is the female. Now if you see here, after making these combinations, what you find, that this would be round and yellow, this would be round and yellow, because Y dominant, R round is dominant. So again this is round and yellow, this is round and yellow, correct? So in this way if you make, you will see that these are all round and yellow, these are all round and yellow. How many? 1, 2, 3, 4, 5, 6, 7, 8 and 9. These are round and yellow. Now for this part you check what is it. This is round and green. This is round and green, and one more you will get round and green. Yes, and the last here we can make a triangle. What are these? This is wrinkle and yellow. Wrinkle and yellow. How many will get? 3. And then this one is your what do you call, wrinkle and green. So in that way, when we observe the phenotypic character, you find a round and yellow 9, round and green 3, wrinkle and yellow 3, and wrinkle and green 1. So this is the phenotypic ratio. Now when we talk about the genotypic ratio, then it is little difficult to find out in that way, no? Because now capital R, capital R, capital Y, capital Y, how many? Only one. Now capital R, capital R, capital Y, small y, if you count how many, let's say this is one, any other, this is another one, like that you have to search. Any more? Suppose say here 2, like that it is difficult. So we can go for something else. What we can go for, in the monohybrid cross we had got the genotypic ratio, if you remember, 1 is to 2 is to 1, isn't it? So what we can do, we can go for a multiplication, because it is dihybrid, so one more you will add, 1 is to 2 is to 1. So what will be, you multiply it like this. So 1 into 1, 1, 1 into 2, 2, then again 1, next with 2 we will do next 2 into 1, 2, 2 into 2, 4, then 2, and again with 1, 1, 2, and 1. So this becomes your genotypic ratio. Similarly, actually phenotypic ratio also the same way it has come, because phenotypic ratio is 3 is to 1, if you remember. So 3 is to 1 again for dihybrid. So then if you multiply by 3, 3, 3, 9, then 3 into 1, 3, with this you multiply now, 1 into 3, 3, and 1 into 1, 1. So this is 9 is to 3 is to 3 is to 1. So that way you can also find out the trihybrid ratio and all. So this is in shape they are showing, see. So this is wrinkled and green, so round and yellow, so these are 9, round and yellow 9, then wrinkled, round and green, you see 3, then wrinkle and yellow, uh, 3, and then wrinkle green 3. So this is dihybrid cross. Now in the dihybrid cross, then what we are able to understand, this through dihybrid cross Mendel explained principle of independent assortment. What happens here? Here we see, we can read it properly. Members of one gene pair segregate independently from other gene pairs during gamete formation. Okay, so members of one gene pair segregate independently. So that means here two gene pairs we had taken, say round and then yellow. So this separation, it can separate independently. Here also this can separate independently. They are not dependent on one another for separation. Since they can separate independently, each pair can separate independently, that is why we are getting different kinds of combinations, isn't it? So this gives rise to many combinations, and this is say one of the main reasons why there are what you call seeing variations, isn't it? You are seeing variations in the offsprings, okay, and this is one of the advantages of sexual reproduction. So last here see for multiple choice question we can see understand this chart. See heterozygous allele pair. Two examples if I take for just monohybrid, I will take single character. Suppose I take TT and I take capital T small t. Now here for the first one, in this, are there any heterozygous pairs? No. In this there will be only one heterozygous pair. Heterozygous means capital T small t, but this is homozygous, not heterozygous. So here no, it is 0. Now kinds of gametes if you see, so we have to write 2 to the power n, where n is representing heterozygous. So if for this it will be 2 to the power 0 means 1, that is why you will find only one type of gamete coming out, and here it is 2 to the power 1 means 2. So what are the types of gametes coming out? Capital T and small t. Similarly, what are the possible combinations? Here they are saying the possible combinations is 4 to the power n. So 4 to the power n means in the first case if it is 4 to the power 0, you get only one combination. That is why whatever be the structure, you will find capital T only. But if you see in the heterozygous one, it is 4 to the power 1 means 4. So what are the combinations you will find? You will find that capital T is there, then capital T, small t, capital T, small t, and then small t, small t. So four different combinations are there. The kinds of phenotype, here first phenotype, it is 2 to the power n, that means 2 to the power 0. So what is the kind of phenotype? 1. Here only you will find tall, but here it is 2 to the power 1. 1 heterozygous is there, means 2. So what are the kinds of phenotypes you are getting? You are getting either tall or you are getting dwarf. Then phenotypic ratio, this is, so we have understood how to do it. Last slide I have told you. Kinds of genotype, is t to the power n. In the F2, generation, it is t to the power n, where n is representing the number of heterozygous. Okay, so if it is 0 in the first case, so here what it will be, 3 to the power 0, whatever it will be only 1. But in this case 3 to the power 1 means you will get what, uh, 3. So what are the 3? One is capital T, and one would be capital T, small t, another type, and then another would be small t, small t. So you're getting 3 possible genotypes. And then genotypic ratio, of course, that has also been explained, how we will find out the genotypic ratio for dihybrid. And if it is trihybrid, then again multiplication 1 is to 2 is to 1, 1 is to 2 is to 1, and again another 1 is to 2 is to 1 for trihybrid, and that goes on. So from this, from MCQ, a lot of questions could be explained. So thank you. So this was the second part of Mendel's mission. Here is just a problem which you can see whether you can do it or not. How many different kinds of F1 gametes and F2 genotypes and F2 phenotypes would be expected from the following crosses? One is this, the other one capital A, B, B cross, small a, small a, small b, small b. Third one is a trihybrid. Try this. Okay, thank you.